I would like to introduce Charles Warchow to talk to you about using the Biodesy Delta in the early stage of drug discovery.
Thanks, JT. Before we start, I'd like to let you know that I have no financial interest in Biodesy, and nor does Novartis. This is a completely independent review of their technology, and this is something that we did as a collaboration in 2014, a little bit into 2015, and basically I'm expressing that we have no conflict of interest here. So today I'll talk to you, as JC mentioned, about fragment screening with Biodesy's platform, and this is a really unique platform. It's something that's new in terms of its capabilities. It's really the first major platform to be able to detect conformation changes of a protein in a biophysical platform. And we'll describe how we did some fragment screening with them, and also how it correlates to very well-known established methods. So I'll have a few introductory slides covering Biodesy and their technology, and then I'll get into details about the studies that we did at Novartis in collaboration with Biodesy. Biodesy was established in 2013, and again, this technique is novel in that it detects conformational changes of a protein upon ligand binding. And so that's the first thing to think about really carefully, because that's different than most other biophysical methods where, for instance, in SPR, you have a compound or protein that binds to a target on a sensor, and you read the binding directly. So here, you're not reading the binding of the ligand binding to the protein target. You're really looking at the effect of that ligand upon the target. So the protein must change conformation in order for you to see a readout in this technology. And that's a really important point, and I'll emphasize that a few times during the talk. So Biodesy has an instrument that was launched earlier this year at SLAS, and it's a 3D4-well plate-based instrument, has liquid handling on board, and it's amenable to numerous protein targets. I'll show you some examples shortly. Very easy to use, and of course they support with great software, reagents, consumables, and tech support. So another slide from Biodesy. So to give you an idea about the breadth of potential applications, they've worked with a number of pharma and biotech collaborators, and also some academics. And this is a slide from about a year ago, so I'm sure the types of targets have expanded since then. And the types of targets that they've worked with include ion channel receptors, soluble kinases, membrane-associated kinases, intrinsically disordered proteins, in academia, protein interaction collaborations, and worked on the famous oncology target, KRAS. So what is fragment-based screening? So you're probably familiar with that method and that discipline. So fragment-based screening is something that's been going on for about 10 or more years. It's really pioneered by people at Abbott with 2D NMR a number of years ago. And the concept is fairly simple. So you have a limited number of small compounds. You perform some screen, do orthogonal validation with some biochemical or biophysical technique, sort out the true positives from the false positives, determine X-ray structure, and then take a look at where there's potential for optimization, do some chemistry, and then repeat that cycle a few times, and then ideally you have something that's very potent and can take to the clinic and help out with some disease. So the advantages of this is that the libraries are small, and they comprise a very large sampling of chemical space. The libraries are very diverse, so you can have numerous small pharmacophores or chemotypes that represent numerous types of molecules that can be expanded upon. The libraries are very high quality, so the QC is very high, so that means the compounds are stable over time and don't contain impurities, which can be a problem in general archive collections. And again, we're talking about relatively small numbers, generally in the order of many hundreds to some thousands, and that's very, very different in scale than what's typically done for HTS, where you're looking at generally millions of compounds. Molecular weight is small, typically less than 300 Daltons, and typically what you find out of these screens is weak binding hits, but even though the potency is not so great, the ligands are very efficient, and there's opportunity for optimizations. So common methods for performing FBS include X-ray crystallography, which is probably the least used, but it's the greatest because if you get an X-ray hit, then of course it's well defined. 2D NMR is also very common. 1D NMR techniques where you're looking at ligand resonances, meaning the small molecule, is also very common. SPR, very common, and differential scanning fluorimetry is common. And sometimes biochemical assays are used, but often those are compromised by false positives. And if you want some references, there's numerous reviews in literature that you can find. So again, emphasizing that the Biodesy technique changes protein, detects changes in protein conformation. Well, why is that important? So it's very important. So it's a frequent and consequential event. It's something that the cell uses over and over again to enable signal transduction and to communicate throughout the cell. And there's a growing body of literature describing conformational change, including even most recently in the most recent issue of ACS Chemical Biology, discussion about a CDK inhibitor, type 1 inhibitor. There's some other articles in there too that cover conformation for disordered proteins. And there's some other nice discussion, nature methods. There's protein NMR, also X-ray crystallography in relation to protein dynamics. And Jody Polisi at Stanford did a really nice paper report on the recruitment of hepatitis C associated with the 40S ribosomal unit. And he did some elegant studies with TRFRET to measure the conformational changes there. And there's numerous other examples, including, of course, GPCRs are very well known to change conformation, and Kabilka made that very well known and very famous. So here's a really great example. This is another slide that Biodesy put together, and this example shows Abl kinase, a very famous oncology target. And it's known that kinases share an active site, and they have some structural features that undergo conformational change as part of their function. And different inhibitors actually give different protein conformations. So type 1 inhibitors and type 2 inhibitors have cause the protein to conformation in very different ways, and so that's a way to distinguish during drug discovery what your hits are doing in a very simple manner. Show some more examples of that soon. And of course, there's type 3 inhibitors, steroid inhibitors, made famous recently by Novartis, actually. So is very ligand changes in conformation, and that can have a big impact on the value of that inhibitor and also its MOA. So here's another example from Biodesy related to kinases. This is a SRC kinase example, and here they looked at three type 1 inhibitors and 3 type 2 inhibitors, and also some negative controls against SRC kinase, and ran them all at 10 micromolar. This is way above the KD for these compounds, and what you see is that the signals are very different. So type 1 inhibitors have conformational changes of about 10 to 20% max, and the type 2 inhibitors much larger conformational changes in this assay, greater than 40%. And as long as you're at saturating doses of compound, this is completely meaningful. So this says that the protein is actually interacting differently with these inhibitors. So the protein conformational change is different in these type 1 and type 2 inhibitors, and that's very well known and established from structural studies. So this is very powerful. This is the kind of information that would take crystallographers a lot of time to get, and also maybe some 2D NMR would be required. And here, in a simple biophysical readout, you can see that these compounds have a different impact on the conformation of the protein. So that's a really that's a big strength of this technology. So I told you a lot about it, about what it can do and what's important, and so how does it work, and what are the physics associated with this? So this is not fluorescence; it's distinct. Second harmonic generation involves irradiating the sample at 800 nanometers, so 2 photons of red light go in, and the readout is actually at half the wavelength, which is 400 nanometers. And so this is completely different from every other method. So it's not absorbance. We're not looking at fluorescence. Second harmonic generation is a unique optical effect. And the way that it works basically is that it requires that a dye is attached to the protein, and then the protein is immobilized on a sensor surface. And if you have a his tag, for example, which is the most common method for immobilization, that orientates the dye in a very specific location in the Z dimension. And so the dye is basically fixed in space and oriented in orthogonal to the XY plane. And when a compound binds, if conformational change occurs, then the dye moves, and then that's observed in the second harmonic generation experiment. So that's how it works. So a very different mechanism. So label with a dye, the protein is fixed in space, meaning the dye is fixed in space. When compound binds, the dye moves, and then the readout is observed. So some implications of this is that compound interferences are minimal, really. So and that's very important because there's lots of interferences in TR assays, for example. There are false positives in many other assays. In this assay, it's something that you always want to be careful of, but it's very unlikely to happen because, first of all, very few compounds are actually active in SHG. So it's not like fluorescent compound. If you have a fluorescent compound, it will interfere most likely in your assay if it's reading out in the same window. But here, compounds just generally are not SHG reactive. So that's one big strength of the technology. So false positives are expected to be low in this readout. The second thing is that even if it was active, which again is highly unlikely, the optical path is really thin because you're radiating from the bottle and from the bottom of the plate, and the optical path, typical Beer's law kind of calculations say that you wouldn't see it basically, or it's highly unlikely that you'll see anything interfering. So that's another big strength of the technology. So now getting into what we did with them, with Biodesy, we being Novartis, we started thinking about how can we apply this technology. And one of the first things that you have to do is go through the process of selecting a target. So this is a little bit different. So if someone comes to you and says, hey, I've got a target and I want to generate hits in a screen for this, you really have to ask the first question, well, what do we know about this target, and do we know if it undergoes conformational change or not, and is that conformational change important? And most often it will be, but that's important to understand. So that's the first question you really need to ask. And so we find this in general that SHG profiling is best in information-rich settings, ideally, where you have that information. You know that the protein is undergoing conformational change. You know that you have ligands that cause that conformational change, and easily set up assays with their platform, and that you have a valid assay system for measuring conformational change. That's not to say that all of this is a must-have. Frequently in drug discovery, we have very little to no information about our target. We've expressed it, in some expression system. We've purified the protein, and we may or may not have control ligands against that protein. And this is the kind of platform where you can actually answer that question. If can I go look and do a small screen and look for compounds actually that cause conformational change, and then you can further develop those compounds. So the nice to have is that you know a lot about your target, but it's not mandatory. So screening, of course, just reemphasizing it's best if you're going to screen, you really want to know that conformational change is important for your readout, and you want to have that validated. And again, it's not mandatory. The throughput for this system is on the order of about 2,000 wells per day, so that's about 6 384 well plates. So that's fairly sizable throughput for this instrument. So I know we have a lot of people with very different backgrounds here, so I want to go over some of the nuts and bolts type things. So as I mentioned, SHG requires labeling with a dye, and it's very straightforward to do that. So Biodesy has a proprietary dye, and it's typical labeling with an NHSS ester conjugate. So you have the probe, it has NHS ester on it. It's a simple mixture and buffer system to label lysines is one way to do that. And of course, as we all know, is a non-specific mechanism for labeling. There is some evidence that even though it's ideally not specific, that some lysines are more reactive than others, and so it's possible that one lysine can be labeled commercially. We'll give you more than one dye sometimes because it's hard to control the reactivity in an exact 1-to-1 ratio. But what I found in looking at multiple systems and what Biodesy's experience is that this overlabeling question that often comes up is not a problem. And of course, you got with assay development and show that your protein synthesis works well. So the other way to label, of course, is with a malamide conjugate that reacts with thiols. So if you have cysteine in your protein, ideally you have one in a very conformational sensitive region. Then you can also label with a cysteine conjugate. And for the work that I'm about to describe, we did all labeling with the SE conjugate. So we labeled lysines. I would have liked to label with cysteine. We couldn't work that out initially, although we got it to work later, and I'll show you that at the end of the talk. So typical assay, so it's really straightforward, and that's one of the great things about this technology. The physics are all ready to work for you. All the protocols are ready to work for you, and it's fairly easy to set up and to demonstrate proof of concept with. The first step is to generate a plate, which is a custom plate from Biodesy, and that needs to have a lipid matrix coated on that plate in order to capture the protein. So they have some vesicles with NTA conjugate. So NTA contains nickel, and that's how you capture his-tag proteins. And I recall that I think this is described in a paper that went out last year on the screen with alpha-synuclein, so you can look at more details there. And I'm sure Biodesy can give more information about that if you need it. So first step, prep the plate with the capture lipid mixture. And then second step is, of course, you do some washing, which I didn't mention here. And then next step is to load the his-tag protein, wash away unbound protein, read SHG response, show that your protein is there, show that your wells are reading uniformly across the plate. And then the next step is to add compound, washed up in there that I didn't mention, and then you read the SHG response, and that's basically it. And you're looking at an SHG response before and after addition of the compound. So it's a delta that you're looking there before the experiment and after the experiment. So now more details about the study we did at Novartis. So this was a blinded study, and that's really important. So we didn't give them any information on the compounds. We had a lot of information about how they bound and what were true positives, or well-validated positives from a number of assays. I'll describe that more in detail. And we basically gave, we labeled the protein in our labs at Novartis and then provided that protein to Biodesy, and then they developed the assay, including putting the protein on the plate, and then we gave them some positive controls, and I'll show you those results shortly, and then demonstrated proof of concept. So as I described this study that we did, again, we're doing fragment screening here. This is not the first time a fragment screening has been done. So Josh and his team, Josh Lasky is the founder, Ben Murray, reported this in the paper I mentioned earlier, an SHG-based screen against alpha-synuclein. That's a Parkinson's target, if I recall correctly. And you can get more details there. So what we were looking at is really, so we certainly believe that work, and what we'd like to know is not just does it work, but how well does it work, and how can we use the information. And we wanted to compare it to a really robust system where we had 2D NMR data, also SPR data, and we have some biochemical results too. So in terms of methods, I mentioned that we did the labeling. The SHG construct that we used was actually an N15 labeled bacterial enzyme with his tag. And we chose to use the N15 labeled material, which has nothing to do with biotechnology, but that's the NMR construct that we used because we were doing 2D NMR. And the value of using that exact same construct is that you don't have to worry about batch-to-batch variations in protein, and that basically rules out that variable as a source of differences. So that's a great way to compare directly the results. We have the exact same protein in both studies, both NMR and in the Biodesy readout. Labeling with protein with the dye, as I mentioned, we use NHS ester chemistry, labeling lysines once again. And in terms of the screen, we did duplicates, about 1,700 fragments at 0.5 millimolar in an NMR buffer, which had some EDTA in it, and it does not have the detergent because NMR experiments don't like detergent. So here's the results with positive controls. We had actually 5 positive controls and 1 unknown compound. And it turns out that unknown had a little bit of activity too. And this gives you a read on the types of signals you can get. And so this is another area where you have to change your thinking a little bit or broaden your thinking a little bit, because typically we associate response with potency. And this is different. So potency, if you run some compound at a fixed concentration, typically the most potent compounds will give you a bigger read. But it's different here because what we're looking at again is not the binding event, it's the conformational change. So it all depends where the compound binds, and then how much the protein moves is a function of that binding. So here we had 5 different, very different compounds. They all bind in the active site of the same time, but get very different responses. So we've got compounds that range from nanomolar all the way to millimolar, and as you can see on the right, some compounds that are even the weaker ones give really fantastic responses, and the more potent one on the left-hand side, compound 1, gives a minimal response, even though it's one of the more potent compounds. So this shows that these compounds have differential effects on the conformation of the protein. We also ran a negative control, which I don't show here, 2 negative controls, and they were SHG negative as we expected. And based on these results, we decided to go ahead and do a screen. So here's what the data looks like from that screen, typical plot. What's different again is that you see signals, actually which I didn't mention in the last slide. In Biodesy's technology, you can see signals that are either positive or negative, and both of those are meaningful. So again, if the signal can be, the conformational change can be determined or related to the amount of protein conformational change in a positive direction or negative direction. So you have to think about hits in both of those directions. And if they are different, that means that you're doing something different to the protein. So the conformation is changing differently for compounds that induce a positive shift or a negative shift. The other thing to note here is that the data is fairly clean, so we have lots of results around 0, which is exactly what you want. And then some duplicates are more variable than others, and that's most likely due to, although these are supposed to be well-behaved fragments, my guess is that some of them are not perfectly well-behaved. So here's the overall results, and I'll just start out by saying that these were overall favorable to us. I think the screen went very well. It was a very clean screen, so the hit rate was around 9% or so for 3 standard deviations. So that's about what we would expect for this target. And what that tells me is that we don't have a ton of false positives in this assay. If we saw something in the order of 20 or 30%, that would maybe wonder, but clearly we had a fairly decent hit rate. And in terms of comparisons to 2D NMR, we did run all these compounds in 2D NMR. We only ran 277 of them. And we found that 58 of the compounds were NMR positive. And so the obvious question is, well, how does SHG compare? And in this, for this set of compounds, we saw that 24 of 58 of those compounds were also SHG positive. So that's about a relationship of about 41%. 29 of the NMR hits were SHG negative, and this is something that you might be concerned about if you were looking at comparing 2 binding techniques, because if you're looking at binding, you want it to hit in both assays. But again, this is not so worrisome. What this tells us is that these NMR positive hits are different, and they're having a different impact on the protein. The other thing that we noted is in the NMR negative hits, it was fairly clean. So if it was NMR negative, it was most often SHG negative too. And then there were some compounds that were SHG hits that we had not tested, and we did follow a little bit of follow-up there. So overall, the summary here is that the screen was clean, and that we found some hits, and the overlap in SHG hits with 2D NMR was about 40%. So we had one question during assay development. We have EDTA in our buffer, and that's something that Biodesy knows can interfere with the capture of the protein, or maybe make the signal drift a little bit, because it can chelate the nickel, and then that has an impact on the binding of the protein in the well. So we did a follow-up screen, and we took the EDTA out, and the bottom line there is the screen improved a little bit, so we got a little bit higher correlation to NMR, so it's fairly similar. So we found that EDTA had maybe a small effect in these studies. We did go back in the second study and looked at some SHG hits that had not been tested by NMR initially, and we found some additional hits, which was fantastic. It's always good to say that your technique can find some novel hits. So one of the obvious questions is, we know the protein's moving here, and we know that our compounds are inducing conformational change based on SHG. And as with any assay, there are hits that are really clear and really obvious based on the responses that you see and the data that you have, and there are some that are not. And then the tendency, of course, is depending on the number of hits, you might select them all if you want to go broad. And if you have tons of hits and you don't do manage those, then maybe you just pick the best ones. And but here we had a minimum set of hits. We had about 60 compounds that were positive in 2D NMR, and so we asked a question from the NMR data: how many of these are really compelling, and how many of these are most likely weak? And the answer is that there were 2 different populations, really. And then the other question is, could SHG help to clarify or help us understand which hits are better in the NMR assay? So is there any correlation between SHG response or SHG status relative to the 2D NMR data? And the answer is yes, there is. So at the lower left-hand panel, you see a plot of the NMR positive hits and also SHG hits, and there's an enrichment in the NMR hits where there's a clear effect seen in the assay data relative to a compound that is SHG positive. So this says that the SHG hits are more likely to be true and having a bigger impact on the protein. If you look at the NMR positive hits that are SHG negative, so these are compounds that are binding but most likely not changing conformation, there still are hits that have a clear effect, but there's no clear enrichment in those hits when it's SHG negative. So this shows that there's differences in the quality of the data, or it correlates to the quality of the data that we see in 2D NMR. We also looked at SPR data for these hits. And now we're adding another dimension here. The two take-home messages here really are that SHG is fairly sensitive, so we are screening at 0.5 millimolar, so that's a fairly robust concentration for detecting compounds. And what we see in terms of those hits by SHG is that the affinities by SPR are on the order of up to 2.5 millimolar. So it's a fairly sensitive technique as well. And the affinity could actually be even greater than 2.5 millimolar. That's as high as our SPR assay goes. So that's very comforting to know that we can pick up weak markers in this assay because we don't know if we can use in our mid-chem programs as well. The other thing to note is, as I have circled in the lower left there, the difference in the two plots is if you have a or SHG negative hit, that SHG is picking up more of the more potent hits too. So if you look at the 0 to 500 micromolar KD, there's about 10 compounds there so that have potencies there. And if you look at the SHG negative hits, which are not moving conformation, the potencies might be a little bit weaker. So this might suggest that there's some relationship between protein movement and affinity too. That would require more work to show that, but that's an interesting potential consequence of conformational change. So Venn diagrams. I had to put it up because it's how you show this data. It's kind of complicated. We're adding yet another dimension here. We're looking at all hits that are NMR positive and comparing overlap in SHG, SPR, and biochemical assay. And what we see basically is that in the biochemical dimension, which is the dimension that's new to this conversation, we see pretty good overlap. So that means that the SHG positive hits are also functionally active as well, and that's exactly what we want. We want to know that we have hits that are functionally active. And the overlap here between SHG and biochem is the best of the three comparisons. So SHG to SPR, SHG to biochem, and then SPR to biochem. It's all really tight. It's a small sample size. We're looking at differences of about 10%. So this would require more data to establish firm conclusions about. But it's also going in the direction that we would like to see. So that's basically what we saw in our studies. I'll just summarize shortly. But one more experiment I mentioned that we looked at the biolabeling, and these results look really great. Initially, I tried to label protein with malamate conjugate, and I was getting I got a homogeneous conjugate in the end. But when we did assay development with it, we experienced some difficulty. There's a lot of variability, and so we decided to go with the NH ester conjugate. And then after finishing, go back to that and try that one more time. We're able to work out some conditions, basically some buffer changes. We added some detergent, if I remember correctly. And what's noticeably different is that the controls look much better. It's much cleaner with thiol labeling, presumably because the thiol that we're labeling is very close to the site of the moving. And so I would anticipate that if we had the time to repeat the screen, that we would actually potentially see more hits and more robust hits, and maybe a little bit different correlation in the data. But overall, the screen was, we feel, is very successful and very valuable for us. So to summarize, fragment screening and binding to a bacterial enzyme is detectable by SHG methods is one of our key conclusions. That was expected, and it's good that we can say that, of course. Correlation to 2D NMR is about 41 to 48% in two different studies. And again, I want to emphasize that not all NMR hits are expected to be positive in this assay. So again, remember, this is not a binding assay. So if a compound can bind to the target and not induce a conformational change, and that compound will be silent in the SHG assay. So when you're screening, you're really screening for some compound that binds and also moves the protein and changes its conformation. We also saw a potential correlation between qualitative NMR assessments. So we saw an enrichment in NMR hits that had a clear effect. It looked like it was about a 2-to-1 enrichment in NMR hits that had a clear effect over a weak effect when those hits were SHG positive. So that could help us in our selection process in the future if we don't have the NMR or if we choose not to use it. And then the affinities for SHG fragments that we had validated by 2D NMR were in the range, and things well into the millimolar range, so around 2.5 millimolar, maybe a little bit greater. So that was also very encouraging. So we know that we have the sensitivity in the assay to detect things that are weak binders that could potentially be further optimized. And then the last point is that many of the SHG hits that we saw were active in a functional biochemical assay, and ultimately that's what we care about. We want them to be functionally active, and we saw a pretty good overlap between the functional activity and SHG hits. I want to acknowledge some contributors, Novartis colleagues. Numerous people helped out with protein expression, advice, consultation, and constructs. Mike Estefik also helped out with 2D NMR. And then at Biodesy, we had lots of help from Josh Slavsky, the founder, Sam Kitts, Ben Murray, Caitlin Connell, and Margaret Foucault. And this is something that we really enjoyed and we hope to use in the future. So thanks for your time.